Alright, so there is a connection between viruses and cancer, and it seems that 15% of viruses are or will lead to cancer. And this is because in mitosis you have the splitting of cells and replication of DNA. Viruses also take advantage of those same enzymes to replicate their DNA, and so they can cause the disruption of gene cycles, or the cell cycle that controls genes. They can trigger the onset of oncogenes into cancer genes, and so a way to treat cancer then is by vaccinating against these viruses. And so one of these viruses in 2006, FDA approved the HPV virus, which will prevent cervical cancer. Prions are like viruses. Oh, they aren't really like viruses. They do cause diseases, but they are proteinaceous infectious particles, meaning they are made of proteins. They cause transmissible spongiform encephalopathies, which are basically where the brain starts to waste away. And you can see here's a picture of a normal brain and then a brain with a prion infection. If it's in a cow, it's called mad cow disease. It's called scrapie in sheep, and it's called Creutzfeldt-Jakob syndrome in humans. And in pretty much it's uncurable. In every infection, it leads to the mental deterioration of the brain and death. Basically, what happens is you have a misfolded protein. A protein, so it's not like a virus, which takes takes hostage of the machinery of the cell. It's just a protein, and it causes other ones to misfold as well. What they found is if you can actually delete the protein that might get infected, it will also prevent the disease, and that's what's happening in this figure. You have a you have a mouse that was the normal folded protein was deleted, and they were infected with the scrapie prion, and it was fine because it didn't have any proteins to misfold, whereas a normal mouse would die after infection. Viroids are similar to viruses. They are little small circular RNAs that will go in and infect plants. Over 10 million coconut palms in the Philippines were recently killed through a viroid. And they replicate by themselves, so they don't need, they still need some expression of other cells, but they might use a different type of RNA to affect the gene expression of the plant. Alright, now we're going to move into prokaryotes. Prokaryotes are single-celled organisms, and they were first seen by Antony van Leeuwenhoek, who was the first person to see microbes through the invention of the microscope. Another prominent scientist, Louis Pasteur, was able to disprove the spontaneous generation theory by proving that there was something, microbes, that actually get into substances which cause them to go bad and spoil. So another person in relation with diseases was Robert Koch, who came up with Koch's postulates, and this is a way to determine if a microbe is actually causing a disease, and he worked with anthrax. Basically, what Koch's postulates are is, in an infected individual, you have to first isolate the microbe, in this case a bacteria, from infected individuals, and you can't find that microbe in healthy individuals. Then you have to take that and isolate it in a medium, and then reinfect it into a normal mouse, and it has to cause that mouse or whatever organism to die or to have the disease, and then it has to be isolated again. And it's kind of redundant, but when you go through these processes, then you know for sure that the causative agent is the thing that you purified in the first place. Prokaryotes are very diverse. They're the oldest known living organisms. They are made of cells. They are also the simplest and the most abundant. They were around for over 1 billion years before eukaryotes showed up, and you can see that in this timeline here. Around 4 billion years you had prokaryotes, and eukaryotes didn't show up till about 2.5 billion years. Most of them, however, are undescribed or undiscovered, up to 99%. They fall into two domains: bacteria, also called eubacteria or true bacteria, and the archaea, which was formerly called the archaea bacteria. Bacteria and archaea do have some structural differences. The lipids in bacteria are unbranched and use ester bonds, while the lipids in archaea are branched or unbranched and use ether bonds in their plasma membrane. Bacteria are found basically everywhere except for in extreme environments, and archaea are pretty much only found in extreme environments. The cell wall in bacteria have peptidoglycan. They replicate with a single replication origin, and transcription and translation are unique. Whereas in archaea, they do not have peptidoglycan. Their replication is more like eukaryotes, and so is their transcription and translation. So, with that being said, you can draw the conclusion that archaea is probably is probably closely related to eukaryotes and less closely related to bacteria, and that's the current hypothesis. Now we can split these up or classify them according to their amino acid sequence of proteins. So just looking at the amino acids, or you can look at guanine cytosine content. You can split up their nucleic acids, their DNA and RNA, and then mix them together and see how much of them hybridize. And closely related species will have more hybridization or base pairing. You can look at their DNA and RNA sequences, especially important RNA sequences like ribosomal RNA, or you can look at the whole genome altogether. And based on those things, we've split bacteria into a number of different groups. And that's what these are. So all prokaryotes are unicellular, are made of one cell. Or most are single-celled. A lot of them will stick together and make a biofilm or other association. They are less than 1 micrometer in diameter, and rather than having a nucleus, they have a nucleoid where their chromosome is located, oftentimes associated with smaller pieces of DNA called plasmids. They do not have membrane-bound organelles, and they do have ribosomes, which are structurally different, that are free-floating, not attached to the endoplasmic reticulum. Some of them have a flagella, which is simpler and different in structure than eukaryotic flagellas. Here's a picture of the flagella. You don't really have to know much. This was just to show you what it looks like. You can also classify them based on whether they're photosynthetic or non-photosynthetic, how they move, so motile or non-motile. They can be colony-forming or filamentous. They can also form spores or have different forms of binary fission, and whether or not they cause human disease. And that was probably the first and most prevalent way of describing them. Some basic shapes: you can have rod-shaped, spherical-shaped, helical-shaped, comma-shaped, and all. And then also the way they aggregate, and these are also found in their name. So staphylococci are clustered circular organism or spherical organ. You can also look at the gram stain, and this really, what it does is it separates bacteria based on the amount of peptidoglycan in their cell walls. Peptidoglycan helps maintain the shape and protects it from external environments. And gram-positive bacteria will turn purple when you gram-stain them, and gram-negative will turn pink. The way this works, you stain all the bacteria with this purple-violet, then you add the iodine. The iodine attaches to the peptidoglycan wall and keeps it from being flushed out when you add alcohol. So the gram-negative bacteria will release that iodine, and then you counterstain it with something else with some red dye. So then you have gram-positive are purple and gram-negative are pink. There are also some accessory structures such as the S layer, which helps depending on the organism, in different things, but most commonly is for adhesion. A capsule is the gelatinous layer outside, also for adhesion or attachment, and also protects against the immune system. Pili are found in bacteria that will exchange genes. And here's an example there. This is the peptidoglycan layer, the S layer, and there's the capsule, and these are the pili of bacteria. Sometimes bacteria will go into a dormant state and where they make an endospore, and this is a very protected layer and it contains only a small amount of cytoplasm and then the DNA. And it will stay in that state until more favorable conditions will be found. This is called an endospore. Tetanus, botulism, and anthrax, so those diseases are caused by a bacteria that can form an endospore. Prokaryotes will also have sometimes these large invaginated plasma membranes, okay, and that is usually for helping in respiration, similar to mitochondria, or photosynthesis, similar to chloroplasts. Prokaryotes have different way of metabolizing. They can or acquiring carbon. We acquire carbon by eating, but they can do it by pulling carbon out of the air, and that's what autotrophs do through photosynthesis. You can also have chemolithoautotrophs who don't use photosynthesis. Instead, they use energy from chemical reactions, and those will happen in things like geysers and volcanic and geothermal vents. Heterotrophs are from inorganic molecules. Photoheterotrophs use light as an energy source, not to photosynthesize, but to use energy to break down carbon. And then chemoheterotrophs, and that's what humans are, get all of their energy from chemical substances. Bacteria will transfer genetic material in a number of different ways. Horizontal gene transfer is just from one to the other, not through reproduction. And that can happen through conjugation, where two of them get together. They get together through a pili and then exchange genetic information, or by transduction, where a virus is used to insert DNA, or by transformation, where just a dead cell and floating piece of DNA will be inserted into the bacteria that way. All of them are also involved in processes in archaea. Conjugation, again, is where you have a pili which attaches two different bacteria, and the small plasmid called the F plasmid. If it's F positive, it has the plasmid. If it's F negative, if it's not, the F positive cell produces a pilus that connects them, and then they exchange through a conjugation bridge. The F plasmid then is copied and goes into the new cell, and now both of them are F positive. That's conjugation. Transduction can be generally where any virus is used, packages some DNA, becomes a prophage, and is inserted into the cell. Or you can have specialized transduction, which occurs in the lysogenic cycle. And the imprecise excision or cutting out of the prophage may have some extra genes with it. Transformation happens when you just have a DNA released from some sort of dead or lysed bacteria, and that goes into the other one. And it can also be natural, artificial. Natural is just where it happens naturally, of course, and then artificial is where humans will actually experimentally insert the DNA into the genes. Alright, and we'll stop there, and we'll do one more lecture, just a few more slides that talk about mutations, resistance, and different diseases.